Dear Nobel Laureates, dear colleagues, dear students, dear friends, it's a great pleasure to have the possibility to announce that we have 3 Nobel Laureates in Chemistry 2016 that will present their lectures for us. My name is Jonas Bergqvist. I'm the chairman of the Chemical Society in Uppsala, and it's a great honor to be the host today. We will, before we start the lectures, actually hand out another prize, a prize that we hand out in Uppsala every year for the best student. And it's maybe not as much money as they hand out in Stockholm, but it's a very, very honorable award. And I would like to ask our Nobel Laureates to join me on the stage, and also ask the student of the year. So it's a great honor to welcome Ricardo Fernandez-Terán to get the prize from our Nobel Laureates. And also Ricardo's supervisor, Burkhard Sept, please. That's the money. So please.
So with my warmest congratulations.
So we are on a quite tight schedule. So without further ado, I would ask Professor Leif Hammarström to join me and introduce the first lecture. Please, Leif.
So it's a pleasure for me to introduce Professor Jean-Pierre Sauvage. And he's been called the master of chemical topology, maybe even a wizard. So he's been able to make molecular rings, interlocked, molecular knots, and other structures. But while a stage wizard or illusionist has a trick to take the rings apart, the real challenge for a chemist is to put them together. And Jean-Pierre Sauvage has done not only that, but he's also shown how to control the motion of these rings in a controlled fashion. And people have then had the idea that you can use this for example, molecular memories, or molecular motors, which is the theme of this year's prize. However, I know Jean-Pierre Sauvage as a spokesman for fundamental research, the importance of fundamental discoveries. I also know him as a strong supporter of young researchers and a very inspiring lecturer. So what to really make of these fundamental discoveries of his is actually up to you young and future researchers in the audience to find out. So please join me in welcoming Professor Jean-Pierre Sauvage for this first lecture today.
Do you hear me? Yeah, very good. It's important, you know, that you hear me. So thank you very much for inviting me in your university. It's a great pleasure. It's a university I have visited already a few times in the past. With Professor Leif Hammarström, we even collaborated, and we published together maybe 20 years ago or so, work which had nothing to do with molecular machines, but which was very much oriented towards photosynthesis and artificial photosynthesis. And I should say that Professor Stenbjörn Sturing was my boss also at that time, because he was the coordinator of a big European network, and we also had great time together. So I'm happy to be here. So let's talk about the field of molecular machinery. And the title of my lecture is, what is difficult, you know, to see the title from here. And it's, you end up with a twisted neck, you know. So the title, I know it by heart anyway. So this is chemical topology, or from chemical topology to molecular machines. And I will start. So I prepared a lecture which is very much history-oriented. And in a way, you know, there is some kind of graduation between my first lecture, Professor Stoddart's second lecture, Professor Feringa's third lecture. I start with the very naive, you know, and simple thing, because maybe our chemistry has been in the same way, you know, quite simple and naive, especially at the beginning. And so the idea was to make topologically non-trivial species, and I will spend just a few seconds on that. Topology is the science of infinitely distortable species. So you can distort as much as you like the object you are handling. If you do not break anything, the topological properties of the species are maintained. They are identical, regardless of the distortions the molecule may be subjected to. And if you look here, you have a ring, and the ring can be drawn in a plane without crossings. So you have a planar graph. So the graph here is planar because it is easily drawn in a two-dimensional space without crossing points. Here you may have exactly the same sequence of chemical bonds, of atoms, but you can notice that the three-dimensional arrangement of the ring has been modified. It's a knot, it's a trefoil knot, and in this case you have a non-planar graph, because you must have three crossings if you want to draw the species in a plane. So it means it's a topologically interesting species, topologically non-trivial. And the first people to introduce these concepts in chemistry were those two scientists, Frisch and Wassermann, and they published a very nice paper in JACS in 1961, which were purely discussion. There was not a single experiment in this publication, but it was introducing topology to chemists. And I consider this is a very important paper because chemists were exposed to topology at some stage, and it was completely new for them in the 60s. Another very important paper, in my opinion, is an Angewandte Chemie paper published in 1964 by Professor Schill and his boss at the time, Professor Lüttinghaus, a legendary organic chemist in Germany. And they were the first to make a catenane. So a two-catenane consisting of two interlocking rings. And that was really a very important event, in my opinion. Beautiful work. And if you check on the internet, you will never find a picture of Professor Schill. And I was lucky 2 months ago, after the Nobel Prize announcement, to receive a picture of Professor Schill from some people who knew him very well, who were professors at the University of Freiburg in Germany, the same university as Professor Schill. So the work was really very elegant, but in terms of quantities you can make, it was somewhat difficult. So catenanes were considered as very exotic species, very difficult to make species till the 80s. And I will now tell you how we became interested in catenanes, and I will try to be as honest as possible. When I started my group, I was much interested in water splitting. And this is our connection with Professor Hammarström, Professor Sturing, and their colleagues, artificial photosynthesis. And artificial photosynthesis was a very topical project, very important field at the end of the 70s because of the first energy crisis. There was a big oil shock, an oil crisis in the 70s, and our financial deciders, you know, the very important people in the various governments, they got the message that other sources of energy had to be investigated, and there was big money for the people working in artificial photosynthesis, and in particular, in trying to split the water molecule to H2 and O2. And one of the stars at that time was ruthenium trisbipyridine. And I know that in this audience, there are quite a few people who are very familiar with ruthenium trisbipyridine, which has very promising, very interesting properties, in particular in relation to electron transfer and energy transfer. But, and there is a but, ruthenium is a very expensive metal. It's a second row transition metal. And we decided, in collaboration with Professor Dave McMillan from Purdue University, who was visiting Strasbourg at the time, that maybe copper could replace ruthenium. And so we started a project on copper complexes, hoping that some of these copper complexes would have interesting properties very much in relation to ruthenium trisbipyridine. And I should say, and I insist on that, David McMillan was the pioneer. He had started at the end of the 70s, and it was when he visited Strasbourg that we started together. So what we did was quite simple. For various reasons, we had prepared this molecule, and it's kind of surprising, but at the beginning of the 80s, it was a new molecule. It was unknown. And so we found some very nice trick to make it. And David saw the molecule at a, there was a poster, there was a small meeting in Strasbourg, and he said, wow, this is exactly the molecule I have been dreaming of for years, but we were unable to make it. But if you make the copper complex of this species, we should obtain something which is very exciting in terms of photophysics and photochemistry. And so the Nobel Foundation recommended the laureates to explain, to talk about the story behind the discovery, and this is exactly that, the story behind the discovery. We made this complex containing two diphenyl phenanthroline ligands. The molecule is nice looking first, but it has very interesting photochemical properties as well, and it led to photochemical reduction of water using copper complexes of this type as photosensitizers. But there was something else. If you look carefully at the molecule, it is pretty obvious that if you are able to connect this point to that point, this other point to that other point here, you make a catenane. You have, you know, your two, let's call that intertwined ligands or entwined ligands, and you can make a catenane very easily on paper, first on paper. And we saw that, and we thought, well, inorganic photochemistry is nice, of course. It's an exciting topic, but making a catenane could be even more exciting, more novel. And we started to make catenanes. I just would like to pay homage to Christian Dietrich Büchecker, who was a great organic chemist, And at the time, she joined my group. We knew each other for many, many years. We were students together. And so we worked together. And thanks to her, who has all the ability, all the expertise for making relatively complicated organic molecules, we started in this field, and it turned out that in a few months, we had the first paper. So after a few months, she was able to make a catenane. We published a paper in tetrahedron letters, and this is something we were speaking about with Leif. It's not a high-impact journal. I'm not sure it is so important to publish your papers in high-impact journals, maybe from time to time. But I mean, for us, you know, it was not that important. And even more, we published the paper in French. And it was kind of paying homage, you know, to my former boss, Jean-Marie Lehn, whose first paper on cryptans and cryptates, published in 1969 with Bernard Dietrich and myself as a co-worker, was published in tetrahedron letters and even more in French. That was a bit of a joke, so to say, but so it forced some people to read French. I'm sure that, you know, the title doesn't need to be translated anyway. So now let me explain the strategy, which is kind of simple. As we have seen before, if we start from the copper complex we had made, we will follow this line here. This is the entwining strategy. You make this complex, you make a ring here, you make another ring here, and at the end of the day, you obtain your catenane. Very, very simple. You can be a little bit more sophisticated. You start from the ring which is here with a coordinating fragment, and this coordinating fragment will attract the transition metal, which will be happy to jump here, and at the same time, this open chain fragment will be threaded through the ring, and you obtain this intermediate, which you can convert to a catenane very easily in a single step. So let's look at the molecules. Again, this is very old work, and the drawings are also very old. They are not ChemDraw, you know, in 83, 84, ChemDraw was still something which the people didn't even know the name of. And they were made by a little bit of Indian ink and a lot of sweat. You know. And that worked fine. Christian Dietrich was making the molecules, and I was in charge of the drawings. So a good agreement, a good arrangement. So we start from this very simple molecule, which can be made at the 10-gram scale very easily in a few days. You mix with copper, and you obtain exactly what you had expected, this molecule. And now you go on. I think you go on. You make the catenane from this intermediate here, just by cyclizing from here to here, from here to here, exactly as what we had planned, and you obtain a 27% of your catenane. At the beginning, 27%, we found it somewhat satisfactory, so we could make maybe 500 milligrams of this molecule in 2 weeks, let's say, provided you're a good organic chemist, of course. We got an X-ray structure of the compound, of the copper complex catenane, which is represented here. And you see that you have a very compact structure, globular structure, with lots of pi-pi stacking interactions. Once you demethylate using potassium cyanide, you obtain a completely different structure. The two rings are now quite remote, or let's say the two phenanthralines are remote from one another. The two rings can glide freely. I mean, that has been shown by many various techniques. and we obtained, of course, a totally different shape. And we said at that time that the molecule underwent a complete metamorphosis. Just to show you why we became interested in catenanes, the first reason for making them is that they were not unknown, but there were no good methods for making them. And of course, it's a challenge to a chemist when a molecule appears to be unknown or almost unknown, and you have an idea for making it. But in biology, there are lots of catenanes and knots everywhere, as you probably know. Proteins, DNA, even viruses form catenanes, and this is routine. I mean, they are not exceptional, they are routine in biology. Now, a little bit of history. So for some time, I mean, there were very few people interested in catenanes. At a very early stage, Fraser Stoddart and his group also published beautiful work. They did very, very nice work based on organic templates, which were mostly pi-pi stacking and hydrogen bonding. Hydrogen bonds were used by two groups in 1992, basically at the same time and leading virtually to the same molecule. Chris Hunter at the University of Sheffield and Fritz Vöckle in Bonn in Germany. Makoto Fujita also produced some very beautiful work, more coordination chemistry oriented with palladium to nitrogen bonds. And that was, I should say, the few groups, let's say the nucleus, really. And out of that, I think the field started to develop very fast. And as you know, nowadays, there are many people interested in catenanes. Just a brief mention of some pioneering work done by Professor Hiroshi Ogino in Tohoku University in Sendai in the north of Japan, based on cyclodextrins. And they could make nice rotaxanes using cyclodextrins and various organic threads. I know that the main topic of the Nobel Prize is not really chemical topologies or chemical topology, but I think, you know, just in 1 minute, I mean, since we like this field very much, I would like to introduce you to some of the topologies which have been made. So that was the very first one. This one followed in 86, 87. The big challenge was making a trephine knot, and we succeeded in 89, and we got an X-ray structure in 1990. And the doubly interlocking catenane was made in 1994. And I should say that the record, I'm not sure Fraser is going to talk about that, but his laboratory produced the most impressive piece of work in the field of topology, and this is related to the Borromean rings. But Fraser, you're not going to talk about that. Yeah. So I can take the liberty of saying that this is the most impressive piece of work. Now let's talk about molecular machines. Molecular machines are everywhere in nature. So if you look at biology, in biology, again, you cannot think of any important biological process not involving molecular machines or motor proteins. You know, they are more known as motor proteins. And I just have two examples here. Atp synthase, so everybody knows now that Atp synthase is a rotary motor. I think there is another aspect of this enzyme which I like very much. It is basically the same enzyme since the beginning of life, 3.8 billion years ago, with archaeas, with very primitive organisms. and mammals, let's say, if we consider that mammals are the most elaborated living organisms. And another one, which is also remarkable, this is kinesin, working on a microtubule in the cell. And kinesin plays a very important role in transporting matter, transporting molecules within the cell. So what about catenanes and rotaxanes? Catenanes and rotaxanes are clearly very well adapted to large amplitude motions, like gliding a ring along an axis, or pirouetting or rotating a ring about the axis on which it has been threaded, or pirouetting or swinging a ring within the other ring in a two-catenane. Again, some of the most important people in the field of catenanes and rotaxanes in motion. So again, Fraser, Vincenzo Balzani, Professor Harada in Osaka, who used cyclodextrins, David Lee, who was a former student of Fraser, and many other people. And Ben, I would like maybe to apologize, but I'm just talking about catenanes and rotaxanes, of course. And I have in other lectures, I have also some slides to talk about molecular machines which do not refer to catenanes and rotaxanes. So let me show you the very first system we made many years ago, and it's a catenane in motion. So we start from a very simple catenane here, similar to what we have seen before, and it is complex to copper 1. And you know that copper has two main oxidation states, copper 1 and copper 2. Copper 1 here is tetrahedrally coordinated. So you have 1, 2, 3, 4 nitrogen atoms in the coordination sphere of the copper 1 center. So copper 1 is very happy. Now we will oxidize copper 1 to copper 2 and generate a D9 electronic configuration metal center. Copper 2 is very unstable, is very unhappy, so to say, with a tetrahedral arrangement, and it prefers 5 or 6 coordinate situations. And thus, the ring, which is here, with its 3 nitrogen atoms in this fragment here, will start to move, it will rotate, and the bidentate chelate, which is here, will be kicked out and replaced by the tridentate terpyridine, which was originally lying at the outside of the molecule. And thus you generate this species with a 5 coordinate copper 2 center, and then the complex is thus very stable. Copper 2 likes it to be coordinated to 5 nitrogen atoms. Now you can go back, you reduce copper 2 back to copper 1. This is very unstable because you have too many nitrogen atoms around copper 1. The ring again will pirouette, and you will regenerate the starting form of the species. I would like to make it clear, there is no control of directionality in this case. It is mostly a pirouetting motion, a 180 degrees motion, but it can go in one direction or in the opposite direction without any preference. This is very easy to monitor because you start from a deep red complex, you generate a deep green complex. Here you have a pale yellowish greenish complex. Here it's too fast, we cannot see the color, and we are back here to the deep red complex. And I should say that the system has been enormously improved in terms of kinetics by modifying the structures of the molecules. We could convert minutes into milliseconds and seconds into microseconds. Just a quick movie here, which was made by Sylvestre Bonnet, a student of mine who was very good at computers, and so he made a nice movie, I think. So we start from copper 1, and you see that you have your 4 nitrogen atoms. The tridentate chelate is lying here. Now let's oxidize copper 1 to copper 2. Copper 2 now will force the system to rearrange, and we have now a 5-coordinate copper 2 complex. You can go back, reduce copper 2 to copper 1, the system will go back to its original situation, etc. So you can do it as many times as you like, in the computer, but surprisingly or not, we could also do it as many times as we liked with the molecules. So we could really switch. We let the system operate automatically, and we could switch many, many times, I don't know how many times, without any sign of degradation. Yeah, I will be on time. I will just finish up by telling you that we have been much interested in trying to make molecular muscles, and in the years 2000 and 2000-something, we made a molecule based on similar concepts, similar ideas, which behaved as a muscle. If you think of muscles, striated muscles, you only have filaments which glide along one another, as you probably know. So you have the thick filaments at the center, the thin filaments at the outside, and when the muscle contracts, of course, you shorten the distance between those two Z disks, and you just have filaments which glide along one another while consuming ATP. And we wanted to make something similar, but we will have no time to discuss all the chemistry behind the work. Just to show you, we made a molecule of this type, which is a rotaxane dimer. And this rotaxane dimer can be moved using chemical signals. We could contract this dimer, we could elongate it, and that also was reversible, just to show you the principle. So now let me conclude. We thought that transition metals were very interesting species to use. And in fact, my group is more a coordination chemistry group rather than supramolecular chemistry. But this is not so important. And so the transition metal was used as a template, which allowed us to make catenanes, rotaxanes, knots, and very many species of this type. And in addition, thanks to the presence of a transition metal in the molecule, we could take advantage of the electrochemical or photochemical properties of the molecules to set them in motion, and thus to make swinging catenanes, molecular shuttles, 2 or 3 station molecular shuttles, wing flapping rotaxanes, but we had no time to talk about that, etc. Let me finish up by thanking the people who've done the work with me. So I should say we had several topics in my group. Again, artificial photosynthesis being a big thing also. So some of the people here worked on catenanes and molecular machines. I paid homage to Christian Dietrich Büchecker, who passed away 8 years ago, very, very sadly. Jean-Claude Chambron, Jean-Paul Collin, Valérie Heitz, Jean-Marc Kern, who died in an airplane crash, Sharm el-Sheikh in 2004, and some younger contributors. But these people were, at some stage, permanent people in my group. The 2 PhD students who started with me in 1979 or so had a very important role, a big impact. I think that the first PhD students you work with Of course, there are many, many other people who joined the group and who worked in this field. I'm afraid we have no time to name them. The same holds true for the molecular machinery field. The Aude Livoret was the first to make a swinging catenane, and Maria Consuelo Jimenez made the first molecular muscle. I also would like to thank my university, the CNRS, the French National Research Agency, the European communities who were extremely generous. And again, I think without the European communities, it would have been basically impossible to do all we wanted to do. My new institute, because I had to move 5 years ago after I retired from the CNRS. I also would like to thank Northwestern University and Fraser and his colleagues because I had at some stage difficult time when I had to retire. My mentor and friend, Jean-Marie Lehn, who had a big influence on my way of tackling research and also on my way of talking to the people or listening to the people in my group. Malcolm Green, the organometallic chemist with whom I learned a lot of transition metal chemistry. Two teachers who had a very important impact on me, Guy Ourisson and Raymond Weiss, and finally my family and my two other friends, laureates, Fraser and Ben, without whom I wouldn't be awarded the Nobel Prize for sure. Thank you.
Thank you so much. And
Thank
we
you.
will, in order to keep time, we will
Sure.
save all questions until the end of all the lectures. So please keep your questions in mind and remember them. So now it's my great honor and pleasure to introduce the next lecture, and it's going to be given by, I thought it would be up there. Sir Fraser Stoddart, which is our next laureate. I think it will maybe come up. Anyway, Fraser has had a fantastic career, and I read a bit before, but also we had the pleasure to meet you last night. And I understand that you have been shifting around a bit with different fields, and also with a very high driving force, actually, to reach out to the younger students. And you did that in a fantastic way last night also. So, without further ado, Fraser, please join me on the stage. And now we will hear something about the fantastic molecules that Fraser has synthesized, rotaxanes and other things.
Well, good morning, and thank you very much, Johannes. I must apologize. I'm not usually holding people up in a presentation like this, but I've actually been violently sick from about 2 in the morning until about 9 o'clock. So if you can make an allowance for me, I'd be happy. That apart, I'm of course absolutely delighted to be here, and it was probably around about 2001 that I made my last visit to Uppsala University. This was with my wife, who was then still alive, and so it was one of these very memorable occasions where we were hosted in a way that I will never forget. The first thing I want to say is that in receiving this Nobel Prize, one of the most heartfelt things is to be sharing it with Jean-Pierre Sauvage and Ben Feringa. Last night, at the closing reception at the Nobel Foundation, the president rather jokingly said that the 2016 class, and this included the physicist and economists. Of course, we didn't have anybody from the literature scene. That we had been one of the best classes ever. And so I saw the physicists got on like a house on fire. What can I say about the chemists? We've been, I think, in a mutual sort of love-in for with Jean-Pierre Sauvage, because we're both a little older than Ben, for at least 35 years. And I've known Ben for about 25 years, and so has Jean-Pierre. Okay, so into the theme of my talk, which is played on the actual citation to some extent, the design and synthesis of molecular machines based on the mechanical bond. And okay, so I will go through the first few slides quite quickly because they reiterate much of what you heard in a beautiful way from Jean-Pierre Sauvage, speaking English in that just characteristically French accent. I always say, you don't have to be able to actually put your eyes on Jean-Pierre. You just have to hear this beautiful accent and you say, this is Jean-Pierre Sauvage. Yeah. All right, so he's introduced you to catenanes and retaxanes. I just want to point out that we use this term mechanical bond because Professor Frisch, way back in early part of the 1950s, coined that term to describe either a catenane or a retaxane at its simplest level. Now, looking at, first of all, retaxanes, this is one of the examples of what I call a mechanically interlocked molecule, or a MIM. Now, if you want to make it easy to write about your chemistry and perhaps even get other people to pay attention, it's quite important to get 3 or 4 letter, particularly 3-letter acronyms. You're all well familiar with DNA, of course, and RNA, and you're all well familiar, I would think, also with metal organic frameworks, MOFs. So a retaxane, according to Wikipedia, is a molecule, and I want to get this point across. Although there are component parts, the blue part becomes a component, and the green and the red part becomes a component. And you can see it's built up on a derivation from Latin. And if you are thinking about molecular machinery, you can very quickly make this association with a counting device that is still used in China. Some of my Chinese graduate students and postdocs are well familiar with the use of the abacus in their training in mathematics. A catenane, again, is also a molecule and derived, once again, from Latin. And I put up this symbol, which I think is familiar to everybody, particularly after this summer, listening to what was happening in Rio, the Olympic symbol, because it was in the mid-1990s that we made a 5-catenane with these different colors picked out in the colors of the Olympic sign. But I want to simplify it by going to the next slide to point out that the 2 rings that are the second and the 4th ones in the series are the same. They're crown ether based with dioxynaphthalene units. And it is these pi electron rich systems that interact with the bipyridinium units in the cyclophane, which is carrying 4 positive charges. This is the same constitution. This is a slightly bigger constitution because we've got to be able to put 2 of these naphthalene units through these units. And, you know, I have to say, in this design, we were so lucky to be able to get the dimensions that you need, namely about 3 times 3.5, which is 10.5. These numbers are a little less, but to allow us to make pi-pi stacking interaction, very similar to what you get in DNA. Also important, not shown in this image, are CHO hydrogen bonds. And finally, CH-pi interactions, which are present in proteins. And so the point I want to get over here is that although we've used quite unnatural building blocks out of the chemical toolbox, the interactions, the molecular recognition is exactly the same as that you find in naturally occurring compounds like DNA and proteins. And we continued this theme and got to a situation where we put an extra ring on here and here and made a branched 7-catenane. I think that still remains one of the highest molecular weight catenanes that has been fully characterized right through to its X-ray crystal structure. What I want to say here is that this work was done by a postdoc who's been with me in Stockholm, David Bellino, and I just want to use his name to illustrate the relationship between Jean-Pierre and I. So after a 3-year postdoctoral period at Birmingham with me, David went and spent a 1.5 years in Strasbourg. In much more recent times, and Jean-Pierre referred to this, he, after he, in quotation marks, retired, he spent 2 wonderful years coming back and forth between Strasbourg and Evanston, Illinois, to interact with myself and my graduate students and postdocs. And I did a check. There are, I think, 10 postdocs and 8 graduate students who appear their names on 7 publications that came out jointly between us. So that gives you some idea of how close we were and still are. Jean-Pierre introduced Ed Wasserman as the person who, in '61, got chemical topology launched. I just want to mention that in the year before that, he had taken these esters, diesters, and had made and used deuterium labeling a catenane in very small yield. And one can argue about the size of this yield. That doesn't matter. You either believe in it or you don't. The point is it's a statistical synthesis. It just relies on the event of a threading of a linear molecule through a cyclic one. There was no, if you like, really positive molecular recognition here. Maybe some van der Waals interactions, but no more. And then again, Jean-Pierre mentioned Gottfried Schill. This is the last 4 steps of his 22-step synthesis using directed covalent bonding in order to complete this bringing together of 2 rings that it's only after cleaving this bond and the acetal here that you get the free rings. Of course, this was a very elegant piece of work in 1964. I think the downside is that once you've broken the covalent bonds, the 2 component parts really don't speak to each other. You would have to reform the covalent bonds. But this has been, to some extent, in my early days, one of my Bibles. So here you have Jean-Pierre again. You have heard in a much more elegant way, using this wonderful intonation that he has to give to the English language, the making of catenanes, and there's the 27% that he extolled the virtues of as a strategy A. He hid from you the fact that at the same time in that wonderful tetrahedron letters paper, by a strategy B, he was able to get a 42% yield. And of course, these yields have just gone through the roof since. We introduced donor-acceptor templation after a decade of investigation into molecular recognition involving systems that became bipyridinium in nature. So once you have 2 positive charges, this is a good electron acceptor, and the hydroquinone rings here, of course, are good electron donors. In the beginning, the dication here in no way recognizes the template. You have to have the formation of the first carbon-nitrogen bond for it to happen. And then I show that it does happen because what we see here is the non-covalent bonding interactions of a CHO and a pi-pi stacking nature being built up, which is then optimized when the second carbon-nitrogen bond is formed. And what was really amazing about this discovery was that in the first time we did it, that at the first time it was a 70% yield. And with a little bit of tweaking, you could get it up to 99, 100%. Well, this was a major breakthrough in our lab. And just to tell you how the friendship between the Sauvage and Stoddart groups is expressed, I did not wait, I did not have to wait to hear from Peter Gurlitz as editor of Angewandte Chemie about either the acceptance or rejection of this paper. I had a telephone call from Jean-Pierre Sauvage, extolling, the virtues of having made it. Right? That's the kind of person he is. Okay, so let's move now to a rotaxane, and in the first instance we'll make a degenerate one. So this is the same sort of approach, except in this particular situation we do it in one pot using these reagents. The same principle, we create an acceptor here which will interact with this template in this sort of way using the same non-covalent bonding interactions that I made you familiar with to get through to a 2-retaxane that has 2 recognition sites. Now, if I redraw, incidentally, it was with a slightly smaller yield, of course, because we don't have the same level of organization here. This is just organized on the slide. It's much more flexible, of course, compared with the previous template, which was cyclic. And so I redraw it, and you see we have a degenerate situation where this ring can jump back and forth between these 2 sites, as shown here in this movie. And at room temperature in deuterated acetone, because this is an NMR experiment, you can roughly see that this is happening 2,000 times a second. Okay, so this was, again, another major breakthrough for us in '91. But in order to take it that step further, we had to make the 2 sites, recognition sites, on the rod portion of the dumbbell different. And so you see now we have introduced here a biphenol and also a benzidine site here. So the red one, biphenol, and the green one is benzidine. And in order to get this ring to move, we have to use some chemistry. So we alter the pH. In other words, we add protons. And because the ring has 4 positive charges, Coulombic repulsion will dictate that the ring goes and sits on exclusively the biphenol unit. And you see this happening here in this particular movie. As the electrons are taken out, the ring goes and resides on the biphenol unit. Okay, so I will fast forward now. This was work that was done in Birmingham in the UK. And what you see here is a cartoon-based representation of the synthesis of a retaxane that has, as the green unit, a tetrathiafulvalene unit, and this is a dioxynaphthalene unit. Because this unit is the more strongly binding of the cyclophane, this is where the synthesis is templated. And now what you see is the moving back and forth as you take electrons out of the TTF unit and make it positively charged. Now, if we take a chip here and create, and when I say we, this was work done in collaboration with Jim Heath, now at Caltech, 4 wires, so we just keep it quite simple, made of polysilicon. This chip goes into a Langmuir trough, and in the Langmuir trough, these are pressurized to a particular point where they're all lined up like soldiers. And then you lift the chip out of the water. So this is called the Langmuir-Blodgett technique when you do that. And you have a monolayer of the molecules sitting on these 4 wires. Now, this represents any one of these about 5,000 molecules because these wires are about 100 nanometers in their width. And this is a 4x4 molecular electronic device. Going fast forward to 2007, Jim Heath was able to do wizardry on a scale that was just mind-boggling and get the wires down to about 16 nanometers wide. And at that stage, this has about 100 bistable retaxanes trapped between each of these crossbar points. And there's the particular retaxane that was used, the tetrathiafulvalene unit, the naphthalene dioxy unit, a hydrophilic end and a hydrophobic end so that they could be put through the Langmuir-Blodgett technique. And this is a very, very large, of course, crossbar device, 400 by 400 wires, and it was tested in parts with a very high threshold, about 1.5 on this scale here for the current ratio for a good switch versus a bad switch. So it's only the ones above 1.5 here that are put in the good category. And to cut a long story short, only about a quarter of the switches, for one reason or another, end up being good. And so this gives us molecular random access memory at a density of 10 to the 11 bits per centimeter. And to push home this achievement by the Heath group, I show you that he was able to use the ASCII2 code to print out using ones and zeros California Institute of Technology, CIT, on this device, which I'll point out to you in an experiment here where in ethanol, white blood cells were suspended above it. The diameter is less than the diameter of a white blood cell. And at the time, and I don't know that it's changed, this is roughly where the semiconductor industry expects to be in, I guess, 40 years from now. The one drawback about these switches is that they are not robust. They might switch about 100 times, sorry, but that's about it. They shake themselves apart. And so I always like to give very up-to-date results in a lecture, and so just recently there was published in JACS this work that puts the switch based on a catenane with again a TTF unit and a dioxynaphthalene unit into the pores of a metal organic framework called NU1000 that's been brought onto the scene by my two colleagues here, and we use the technique that they have popularized called SALI, or solvent-assisted ligand incorporation, which allows us to plug in this unit here in the last step of a making of the molecular electronic device. And here you can see on a thin film, it turns out, the switching back and forth between an on and an off state. Okay, so before I leave switches, let me just mention that also in collaboration with an ex-colleague at UCLA, Jeff Zink, we have covered mesoporous silica nanoparticles, roughly 100, 150 nanometers in diameter, with cavities that are roughly 2 nanometers in diameter, sufficient to incorporate drug molecules, anti-cancer drugs, and to use two types of what we call valves. There are ones that are based on ritaxanes that have two sites on them, and so you have the switch, and there's another one that's based on just simply taking off the stopper here and allowing the ring to leave, and we call that a snap top. Here's just a movie showing what happens during this process. So the drug is put into the mesoporous silica nanoparticle under a concentration gradient, and then the nanoparticle here will find itself, hopefully, in the presence of a diseased cell. It, by epitosis or whatever, makes its way inside, and then you see the opening of the pores here as the rings move, and of course this, in principle, is bad news for the cell. All right, so finally, just to go to say something about our more recent work on molecular machines, and what I've got to get over here, and fortunately Jean-Pierre, and even more so Ben Feringa, will relate what we're doing to the zillions of molecular machines that are present in our bodies. And of course these machines work quite differently. We have no good reason to compare their mechanism with those of macroscopic machines that we're all well familiar with. And that difference lies in the juggling about of energy barriers, so that you see a dumbbell with a green and a sort of blue stopper at the end here, and in order to take this ring, which you're now familiar with, the cyclophane, and bring it onto this recognition site, a dioxynaphthalene unit, we will go over the neutral end here because charge repulsion will stop it from coming over the right end, and it will, under this directionally controlled threading, because of the charge difference, it'll mount this barrier here, as we'll see for these little rings in a minute, because this is a lower energy barrier. And then once it gets onto this site, and we're at this point here, if we now reduce the system, and that is go for the tetracation to the bis radical dication, then we cut down the electrostatic repulsion, so it makes it easier to go over here, and we remove the molecular recognition, and we also mysteriously make it less easy to go over the neutral site. So what you see is a raise in this well and a diminish energy for the transition state here. And so we can move this molecule from left to right, as I'm showing here in this movie. Well, here is a little touch into nature. We're doing something that's very similar to what you see in bacteriorhodopsin, when protons are pumped from one side of a membrane to the other through this membrane of, obviously, alpha helices. the actual constitution of the dumbbell that I showed you is shown here with this dioxynaphthalene unit, this isopropyl phenol, and this pyridinium unit with 3,5 substitution here with methyl groups. And I should say all of this subtle substitution is very necessary to get this system to work the way I've been showing you. And literally, much computational work has to be carried out. And on top of that, not single, but tens, if not hundreds of trial experiments have to be performed. And so what happened was that in 2010, we discovered another form of molecular recognition that had been waiting to fall into our lap for 25 years. And when it did, I just thought, well, how stupid we were not to do it earlier. But therein is the reason for doing research, that sometimes things are sitting there waiting to be done, and it takes somebody to come along. And this was Ali Trabolsi, who was trained in Strasbourg as a physical chemist, I should say, had the courage to go to a glove box and do the chemistry that you see here. In other words, he used zinc metal, as it turned out, to put 3 electrons into this 6 positive charge assembly to find out that we got this massively stable complex between a radical cation and a bis radical cation, and the energy that is involved here is quite considerable, of course. And the attraction is under reduced conditions, but of course, when we oxidize, we'll get enormous repulsion because of putting back the 6 positive charges. And so we were able to conceive of a situation with a slightly different design now. I've turned the dumbbell through 180 degrees, the charge components on the left, the neutral ones on the right, and now we have not a naphthalene, but a bipyridinium unit here. And the way we go through this sequence is by bringing it on from the left when it's reduced, and then it relaxes, it comes into this well here, and then when we oxidize, we raise the energy level enormously because of Coulombic repulsion, and it leaves from the right-hand side. Just parenthetically, we used this radical templation to do something that had never crossed our minds during about 25 years working with this cyclophane, and we could use these radical pairing interactions to make this tetraradical cation as a radical species, and an oxidation. It produces this persistent organic radical. It stops at the septacation with 1 radical in it. And you can get that at 41% yield. And this homocatenane was described by one of our postdocs from the past as an intellectually disruptive discovery. Eventually, we were able to get conditions which could take it up to the octacation, as you see here. So here you have in 1 nanometer an amazing molecule with 4 positive charges in each ring, i.e. 8 positive charges overall. Now, returning to rotaxanes, we took this long thread here with a bipyridinium unit in the middle, and the blue box, if I might call it that, and used chemistry that you heard already from Jean-Pierre Sauvage, the ruthenium bipi, as a photosensitizer in the presence of a sacrificial electron donor to make the tris radical trication, and then some click chemistry to put these stoppers in place. And having done that, of course, in a glove box under an inert atmosphere, when we brought it out to air, we found that the ring moves away from the recognition site because of Coulombic repulsion. So this set up the scene to be able, and here's the cartoon, I'll show you the structure in a minute, to design a molecular pump. So what we're doing here is bringing the ring on from the left because of the radical recognition site when it's being reduced, and you see this happening in this movie here, and then forcing it over by dint of Coulombic repulsion, what I'll call a speed bump, onto a collecting chain. And what's important now is that we're not letting the ring go back into solution, which means that in the early work, we weren't doing any work. We just take a ring out of solution, move it along a rod, and put it back again. Here we're collecting it, and this represents what one needs to achieve in building an artificial molecular motor. That is that you do work and you go to a high energy state. And so with yet another movie here, I show what is going on, namely it comes on under reduction conditions, and then under oxidation it will go over onto that collecting chain. And so what one sees here is the same sort of routine that I showed before with the neutral system based on the naphthalene dioxide unit, but now we have a lot of force because of the Coulombic repulsion to push the ring onto the collecting chain. And the situation is even better when we look at the situation for the coming on of the first ring. The barrier it has to go over here, over the speed bump, to give a 90% yield, which is very welcome, of course, is that number there of 23. And we were very much heartened when we saw that having one ring on here does not alter with an experimental area the barrier that is needed to put the second one on. So this told us we should be able to put multiple rings onto this collecting chain. And this is what we're about at the moment. Again, using this movie, if you like, to show that we're bringing rings on from solution, and as we do that, using reduction and then oxidation, you'll notice that we're raising the energy level of this collecting portion here every time. In other words, we are doing work. And I'll bring on a fourth one and raise it yet again. All right, so that's really the name of the game, doing chemistry away from equilibrium. And this is, I think, in a fundamental sense, what is 2 minutes? Okay, very important. Each cycle traps one ring in a high energy state. Some of the energy input is trapped, work is done. Where could it lead to? It could lead to very interesting new materials, entropically and actually enthalpically dominated polyuretaxanes. And what is going on in the lab at the moment is to have a pump on each end of a long polymer molecule so that we can pump 2 rings on at once, so 2, 4, 6, 8. And that's going extremely well, again, using the photosensitizer that Jean-Pierre brought to your attention. And here's just a movie showing the principle of bringing ring ends on from both ends and stacking them onto this chain. So the point that I do want to hammer home is that we are designing non-equilibrium systems. The motors in our body are not working at equilibrium, they're working far away from equilibrium, and this is really the challenge to we pioneers, if that's what we're going to be called, in the making and realization of artificial molecular machinery. We've got to find out how to do chemistry away from equilibrium. We've got to find out how to do chemistry the way we weren't taught as an undergraduate. We've got to break away from the mold of doing chemistry at equilibrium. So, very quickly, here is an image of what the applications could be. With cyclodextrins and polyethylene glycol, there have already been familiar but important steps taken in Japan by Professors Ito and Kato. Here I just mention in this image on the right-hand side that it's important to ask ourselves how we do design systems that operate repetitively and progressively away from equilibrium. This is a fundamental challenge that is with us today. And so in a nutshell, we've taken a best part of 25 years to go from the work that I described at the beginning, the molecular shuttle, making the molecular switch, and that took quite a lot of time. The early work always does. It went much, much faster in this latter phase where we went to unidirectional transportation, then energetically demanding transportation, and now the construction of artificial molecular pumps. So just to close, of course, Ben Feringa is a leading molecular machinist, as you'll hear in a very, I would say, firm and Dutch way in a few minutes. he holds nothing back, and so should he. But I just want to say what has been the link between Ben and I. So in the United States, we launched, together with Joseph Mika, a symposium sequence or workshop sequence at Telluride in Colorado on making molecular motor switches and machines. And also in 2015, we started the first set of research Gordon conferences together in New Hampshire and Massachusetts. So we are very much pushing the field in other ways, Ben and myself, and of course Jean-Pierre is also very interested in these developments. Other machine people, I won't mention names, have been mentioned, some of them already are shown there. And this is just the young people, in many cases, coming into the game. I must single out Dina Stumian. He's a physicist who has brought a lot of intellectual input into the design of these machines by ourselves and by people like Alberto and David Lee. And finally, a word about, as these come up, about 400 graduate students and postdocs have gone through my group in, I think, 46 years. I wouldn't be standing here if it wasn't for the amazing, absolutely amazing talent of most of these people between roughly the ages of 22 and 32. And that is, of course, more important, but I must not forget to mention that our work has been very well funded, both in the UK, in the US, and more recently from Saudi Arabia by this organization. And so, as these names flash by, and I know I've gone over in time probably, and they want to shoot me, I just want to thank you for the opportunity to come back here and speak to you. It's been a great pleasure.
So, thank you so much, Fraser. Fantastic talk. Absolutely amazing. Chemistry is so beautiful, for sure. So, next lecture. Next lecture will be introduced by Associate Professor Tim Bowden. Please, Tim.
So it's my pleasure to introduce the final speaker of today. It's the Nobel Laureate, Professor Ben Feringa from Groningen University and the Strating Institute of Chemistry. And Ben is hosting a fairly large group of people working on multidisciplinary research. It's a broad spectrum of chemistry and organic chemistry. It's everything from synthesis, homochirality, chirality, isometric synthesis, supramolecular chemistry, switches. It's a long story, but I think what's important here is that all these fields, they cross-fertilize each other. So everyone can learn from each other. And at the heart of the research is the molecule. That's the important aspect of the chemistry, the molecule. So it's about breaking and making bonds, and it's about controlling the molecular revolution. So I leave the floor to you, Ben, please.
Okay, thank you very much, Tim, for your kind words. It's a great pleasure to be introduced by Tim, who was a postdoc in my group, I think about 15 years ago, working on organogels and hydrogels, self-assembly. That was different from machines at that time, but it was a fascinating project. Thank you so much. It's a great honor to be here. Thanks, Uppsala University. There is a special relation between Uppsala University and the University of Groningen, where I am working with the students. And that is, we have a long-standing cooperation and exchange of students. And it's a great honor and pleasure that our ambassador of the Netherlands is here, the president of our university and the rector of our university are in the audience. I'm not sure if they came for me, but it's really great that they are here. Thank you so much. I'm very honored. It's also an honor that my daughter is here, Hannah, who is sitting here at the front row. She was recently here for 5 months, working in the Claesen-Wells group on vascular systems as part of a master's study. So she spent 5 months in Uppsala. And I was here before, 10 years ago or so, once to give a lecture. So it's a fantastic place to be. And thanks you for all coming here. And let me finish with thanking the Nobel Foundation, of course, the Royal Academy, and the Swedish people for giving us such a royal treatment in the past 10 days. It was absolutely fantastic. Thank you so much. So now my time is up, because we are running late, so I can only focus on the very fast motors. No, serious, I shortened my lecture a little bit, but I think I have to speed up. So the art of building small, and I have to go back in time to start, because sometimes I feel like the Wright brothers, as I said also last week, in 1903, when they were flying for the first time, and people said if God wanted us to fly, he would have given us wings. Nobody would have predicted that 100 years later we would have an Airbus built in Europe or a Boeing 747. But realize, although we are inspired by the flying bird and Mother Nature, this is neither a copy nor a mimic of the flying bird. The materials, the 3 million parts, are all artificial, not the soft materials of the bird, of our own body. And the flying principle is different. I would not have been here in Sweden if the bird or the plane was flying like this. So it does not work. But it works perfectly. But also we should be very humble, and as scientists we know that, because 100 years later we are still not able to build a pigeon. We are not able to build a single cell of a pigeon, and not even one compartment of the cell of the pigeon. So there's still a lot of work to be done. So when we are synthetic chemists, like my colleagues here, we build on making molecules, and in the art of building small and doing chemical synthesis, we try to design new molecules or modify molecules and build molecules in the laboratories that never existed before. Simply because we wanted to test the theory, or we want to build a molecule for building a machine or a switch, or for a drug, or for a new plastic, or a new catalyst for our industries. This, by the way, is a catalyst. So, when we look at our macro machines, because I'm going to talk about switches and motors, or about the machines in our body, here you see the artificial robot in a car manufacturing plant, 2 meters in size. Here is the robot in your body, the ribosome, which generates the proteins. It's working very hard in your body, in your cells now. It's 24 nanometers in diameter, and what I want to emphasize, there is an effect of length scale. Here, you realize when a car does not drive, it is heavy, and you have to push it in the garage. It takes a lot of energy to push it. The weight, the gravity, and so play a crucial role. Here, in your body at the nanoscale, at the molecular scale, it is different effects. It is not gravity and weight. It is the intermolecular interactions that Sir Fraser was talking about. Surface interactions, viscosity, molecular effects. So this is the effect of length scale. We work at low Reynolds numbers, and it's not so much about how to get motion, but how to control motion. We live in the world of the Brownian motion, the temperature effects. Molecules move like crazy. How to control motion. So when we look at the machines, we get absolutely astonished by these beautiful machines in your body. Like the rotary motor, I will come back to that, the ATPase, the bio-nano motors that make that your muscles can move, they walk over the filaments, as was alluded to by Jean-Pierre, the ribosomes, the bacterioflagella motor. It's only a small collection of the 50 or so different motors that are in your body that make it possible that we can walk, we can see each other, we can have things go in and out of the cell, that cells can divide, etc. But we started many years ago being inspired by the optical switching in the eye. The fact that you can see me and I can see you is due to this very simple molecular process, which the retinal, the band molecule in your eye, is switched by light to a more linear molecule. It is a rotation around a carbon-carbon double bond. And for those who had first year undergraduate chemistry, you would say, but that's impossible to rotate normally around a carbon-carbon double bond. That's why we need the light. The light hits the molecule, it breaks temporarily the bond. In a fraction of picoseconds, it rotates, it reestablishes the bond, but it has done the rotation. And that triggers then a cascade of events to your brain. And that makes it possible that we see. Not with one molecule, but with millions together of these nanoscale molecules. So we thought, can we make a 0-1 switch and do information storage and maybe computing at the nanoscale? But you might say, why don't you take the molecule from your eye? But when you take it out of the protein environment, it is degrading so easily it does not work. Nature has protected it. So we have to build artificial systems, and so we build these molecules, and you see here a carbon-carbon double bond, and we switch with light of 2 different wavelengths between 2 states. This is the 0 state, and this is the 1 state. Why do I show these screws here? Because this is a left-handed helix, and this is a right-handed helix, just like your left hand and your right hand. The 0 is the left hand, and this 1 is the right hand. And why did we do that? Because we were facing a fundamental problem that we could store information, but we could not read it out without destroying the information because we were using light and it was switching back. So we had to do a trick, and we introduced this so-called stereochemistry of chirality, the handedness of the molecules, and that allowed us to read out the information far outside the regime where you do the switching with the light. And so then we could make information storage systems. And together with Philips, for instance, we worked on that, and we made actually CDs, yeah, where we could store 0s and 1s, write information, and you can store a hell of a lot of information. It never made it to a technical device so far, because we failed to store the high density, because then you need every molecule, you know, to be addressed individually, and we could not do that. In the meantime, in the last decade, we made a lot of molecular information storage system and switching devices together with our colleagues from physics. And few of the time, I will not allude on that. I skipped a few slides, but Professor Stoddart showed you some beautiful examples of his very elegant work. So the field is moving forward there. So what are the challenges in our field? We all know about molecules, but our body and our cells, it's dynamic molecular systems, all these dynamic functions. And as Fraser Stoddart already mentioned, it's out of equilibrium. If we were in equilibrium, ladies and gentlemen, we all would be dead. We are constantly pushed out of equilibrium. And that makes that a system is living. So when we go from the cell, we have to deal with self-assembly, trigger and response functions, and motions. And this in a dynamic sense, of course. Motion is dynamic per definition. And then you can go to responsive materials, materials that change and adapt, just like in your body. Smart drugs, maybe soft robotics in the future, many years from now. And I will show you some examples of how we are addressing this. Let's first go to self-assembly. There is no engineers in your body and your cells that engineer these components. The molecules are instructed, they find their way, they assemble the components and the machinery themselves. So what we did is, for instance, we made amphiphiles, just like the soaps that you used this morning when you took a shower, but now we make them so special that they form nice bilayers or membranes. So, and in the middle of this molecule here, the core part, we put a so-called overcrowded alkene, which acts as a photochemical or electro redox switch. So electrochemical or photochemical switching. And when you put these molecules in water, what they do is, if it works, yes, they assemble in these beautiful nanotubes, completely by self-assembly. The molecules are perfectly instructed to make these tubes. They are 28 nanometers in diameter, 3 nanometer walls, perfect, all equal. Yeah? And you see here we attach these tiny vesicles, these tiny self-assembled balls to it, just like a primitive cell. And this is probably the tiniest reaction tube that you have ever seen in your life. They are extremely stable because these are designed in the way that, like 2 combs, the tails are interdigitating and they form a very stable membrane. So you can take away this ball and put it on again. You can load it up with something like a drug, and you can use it, for instance, for drug delivery. You can also then hit it with light, because although they are extremely stable, you see here a single tube, and we can follow actually when we hit it with light, when we irradiate, you see the process of bending and changing. And this is single nanotubes changing shape. At a certain point they open and material pops out. These measurements were in part done by Jack Byerly, who was a member also of Jean-Pierre Sauvage's team. You saw his name on the slide. So, we also went a step further and we thought, can we make a nano valve that we can open and close? That would be really cute, because then you can think of building kind of more complex machineries, things that can go in and out. So we took a protein complex from the coli bacteria in your body. And this is a side view sticking through the membrane. This is a top view. And you can see this is a complex protein structure which is tightly closed or open. It's like a diaphragm in a camera. And it's normally tightly closed. But then the pressure, the osmotic pressure in the bacteria cell builds up. It prevents itself by opening this gate and material flows out. And this is a way to survive osmotic pressure when the salt concentration, for instance, becomes too high. And so we thought, what happens if we would build in a light switch and we could open and close it with light? So what we did is, and this is a bit of a complex slide, we went to the genome, modified it, so that we could install cysteine groups in the protein complex. These are these blue dots here. We hook up synthetic light switches. We can switch between two states with these wavelengths of light. And then you see here plus and minus. We introduce charges. And we thought, if we have a couple of these switches there, these charges will repel, and water will flow in the pore, and it will open the pore. And so what we did is, after it worked, we reconstituted it in an artificial cell in the wall, and then we loaded up with a dye or with a drug, a pharmaceutical. You hit it with light, and if it works, the pore opens, material flows out. So you open a 3 nanometer pore. So here we take advantage of some material we get from Mother Nature. We re-engineer it, building in this 1 nanometer or smaller light switch, and now we are able to control and materials can flow out. And we can put it in a membrane. So why do we do this? Because we can do controlled drug delivery maybe, or maybe self-healing material. And this might come sooner than you think. Maybe not based on this system, but once you know the principle. You make a scratch. The scratch in your hand heals itself normally when you are healthy. Now think of your car. You have a scratch in your car, in the coating. There are these tiny capsules. Light comes, due to the fact that you have a scratch, light comes in, it opens these capsules, and the material heals itself. And after a day, it's fine. So don't worry about scratch. No, not at the moment, but in the future. This is future. And I think in 10 years' time we have this kind of system. So, the other example I want to show you is problems with precision medicine. We all know when we take drugs all the time, and they go everywhere in your body, and hopefully they do the job there where there is an infection, or there is a tumor, or there is something where it is needed. So we thought, can you put in light switches, for instance, into a medicine so that you can have high precision in therapeutic action? The idea is that you take a drug, and there where, for instance, you have an infection, you hit with a lamp or a laser, and then you switch on exactly there on the spot your drug activity, and it doesn't do anything else in your body, and you switch it on. Does it work? Now, there are two major problems where we were focusing on. Bacterial resistance. It was mentioned by the World Health Organization of one of the humanity's ticking time bombs. We all read in the newspapers about antibacterial resistance. That's our own fault, due to the overuse of antibiotics that we like so much to survive. But there is a serious problem. Second, we focused also on cancer treatment, and you know all the nasty side effects of chemotherapy. So what we did is, for instance, to give you an example, we took Cipro, which is used in the clinic as a broad-spectrum antibiotic. We modified it with a light switch, and then we can activate and deactivate with light. And the whole idea is that you have an infection here. You take this compound, this antibiotic. It goes everywhere. It doesn't harm your normal bacteria in your body. You hit it with a light. It activates it. It does its job. And after 12 hours or 6 hours, or you can fine-tune it, it goes out in the environment, it's inactive, and it doesn't do anything anymore. This is the dream, of course. But we have done this in the laboratory with cells, and it works nice. You can even take a mask, and you can grow bacteria in certain patterns because you switch antibiotic activity on and off. No resistance buildup. That's the idea of we can control biofilm formation, because that's another major issue. So we also use this chemotherapeutic agent, bortezomib, which is used in the clinic for treating several tumors, put in a light switch, and now we can control proteasome inhibition, which is an important pathway there where this drug attacks, and we can switch between an inactive and a very active drug. And that hopefully then will work as follows in the future. Once again, this is a perspective for the future, that with the new techniques of imaging that they are also developed here in Uppsala, the MRI, the fluorescent techniques, the PET imaging, etc., that we get this very high sophisticated detection techniques of tiny tumors, for instance. Maybe it then can be fed to a laser and activate the drug on the spot, and you don't get the nasty side effects. That's a bit of a dream. We all know that it takes a long time to get into the clinic. We are at the early stages. We work together now with cancer surgeons, but you know how long it will take. Maybe we will never make a drug. But then the fact that you can change activity, the most important advance in the future maybe is that we can up and down regulate biological pathways in the complex biological system. Maybe that's a more important advance of making these switchable systems. So there are many challenges, of course, to activate and deactivate and to make high spatial precision and smart pharmaceuticals. The major challenge at this moment is to use near-infrared light, because when you have visible or UV light, you cannot get high penetration depth, but with near-infrared light, heat, you can go centimeters deep and really reach organs. And very recently, in the Journal of American Chemical Society, we published a paper where we can really use near-infrared light for the first time to activate such an antibiotic. And of course, imaging, I mentioned that already. So let me move on then to the motors. How did we came from switches to motors? And this story goes back many years to my PhD thesis in 1977. And in page 61, you see these structures, these overcrowded alkenes that you have seen before, more or less, when I showed you these switches, these light switches, which we try to mimic the process in the eye. And I forgot that for a while, because after my studies in Groningen, I went 6.5 years to Shell to work in industry, which was a tremendous opportunity, of course, to learn. But then I decided to go back to the university to work, build up my own group, and to work with young students and to teach. And then I got this challenge of building optical computing and optical information storage system. That was a bit the idea. And in 1991, we published our first paper about these optical switches. And then, what is going on here? There's no power? Can somebody help me? Because I think I have the power on. Is this not in? Maybe try another one before my computer dies. Okay, and so in 1999, it led to the first unidirectional motor, rotary motor. I will mention it. Motors on surfaces, controlling motion of micro objects, molecular nano car. And in 2016, I came to Stockholm, and I brought this car. And this is the molecular car from Groningen. I will mention in a second. So going to motors, this is an electromotor. This is the Atpase motor in the cell membrane of your body. And you see there is a difference in size in particular. This is 10 nanometers. But the rotary principle, we should not forget, is seen here. This is the Atpase motor. I would argue the most important motor in this world. Everybody has this in his body, the Atpase, because it generates the ATP, the fuel in our body. And I don't know if you realize, I learned this recently, that about 30 kilograms of ATP are produced and consumed in your body every day. This boils down to between 100 million and 200 million tons of ATP produced by these motors every 24 hours in the world. That's quite impressive, by a nano motor. So I hope I have convinced you that motors are important. So this is rotation, and it's rotation in a certain direction. And so how to control rotary motor motion and how to control left and right. That are the key fundamental problems. we were facing. And now I have to go back to the first Nobel laureate, Jacobus van 't Hoff, who was a Dutchman, the first Nobel laureate in chemistry, who taught us that stereochemistry, the three-dimensional structure of molecules, left and right, is really important. And Hans Winberg, who was my professor, a master in stereochemistry. And why is this so important? Because we know left-handed amino acids, right-handed DNA, the basis of the molecules of life. Or in other words, mirror image symmetry is a signature of life. So how can you control left and right? And so what we had to do is to distinguish between left and right and make a molecule that is only right-handed or left-handed, and we can design that in the laboratories. And so we built this, and this is the first unidirectional rotary motor built on the principle that the energy that goes in, the fuel, is from the light, and the rotation is clockwise or counterclockwise, because the molecule is either right-handed or left-handed. And you see again the double bond here. We break it temporarily with light, then it rotates a quarter turn, then it can continue rotating in the same direction. So it's a power stroke motor because we consume energy, we have directional movement, and we have a repetitive process. Once again, think of your motor in your car. If there's no fuel, no movement. If there's no control of directionality, equal probability going backward and forward, you wouldn't get much further either. So we had to control that, and it worked out. So it works as follows: a very fast double bond isomerization, a slow helix inversion, because when you put in energy here, this molecule is rather unstable, and in contrast to switching back, like the molecules in your eye, it was switching in a forward direction, doing another quarter turn. And we found it more or less by accident. This is serendipity in science. Because we were building sophisticated switches, and instead of switching 90 degrees, suddenly we found that it was doing something weird. And when we figured it out what it was, by discussing with the students and doing a number of experiments, we figured out that it had moved on another quarter turn. And when we realized that it has done 2 quarter turns in the same direction, you think, wow, maybe it can do 360 degree cycle. So just to repeat the process. And that's what we did. And it was rotating in a unidirectional sense, as you see it also. So we then redesigned by making a new, we made 50 or so, 60 different types of motors over the years. Because the first one rotated only once an hour. Now that's not much of a motor. You cannot build a car with that. But then in the meantime, by carefully changing the molecular structure, the electronics and the sterics, and in particularly this helix inversion process, so that it continues rotating and doesn't go back. We had to fine-tune those barriers, but by doing that, now we have motors that rotate once an hour, once a minute, once a second, once a nanosecond, and we go down all to 10 million rotations per second. Now I have to be careful here. Because 10 million rotations per second is extremely fast. But in the practice, of course, we have to put these motors in a solution or on a surface or in a polymer matrix. They will not rotate 10 million times per second. This is the theoretical value based on spectroscopic data. But in practice, I think what we find is maybe 3,000 to 4,000 times per second, which is pretty good, I would say. So we build them on surfaces, we build them in liquid crystals. I'll come to that in a second. We can make polymers that we can change the polymer architecture and helicity of macromolecular materials, say plastics, and we could build a nano car. So rotating of a micro object, what you see here is when you take a display material like my laptop, or you take your smartphone and you take away the glass plate, you have the soft material of the liquid crystal display. And this is the surface. You know, it's soft material behind the glass plate. And you see then, this is almost like waves on the sea. And we put a glass rod there. These are the dimensions, micrometer dimensions. 10,000 times smaller is the molecular motor, which is in there. You don't see it, but it is used to reorganize this soft material in the display, the liquid crystal material. And when you now switch on the lamp, this is what you see. You see the surface changing. You see the color changing, you see the object spinning in a clockwise manner. We can spin it clockwise, we can spin it counterclockwise. So this is our way to amplify the effect from the nanoscale to the microscale, but also to make responsive surfaces, because we have these soft surfaces and they change simply by irradiation with light. And what we are currently doing in my laboratory, for instance, to grow cells on top of it. And if you know, stem cells are very sensitive to surface stress and surface architecture. And so what we try to do now is to deviate the cell growth on the surface by having these responsive surfaces. And it's just one of the challenges we try to address at this moment with the students in my lab. So I have to study cell biology now, which is a nice challenge for me. We also make this kind of motors on surfaces, and the real why we do this, because if you ever want to make a device, and this is what Fraser was alluding to, you have to interface them with say, electronic components or with surfaces, etc. So we build legs, the stator, an axle, and we have a rotor. We do self-assembly on surfaces, and then we make these tiny windmills that can rotate, for instance, on gold nanoparticles or on flat surfaces. And we struggled a lot with it to make this responsive surface initially, because the problem was we had these systems too close to the surface. And when you have studied a little bit of physics, you know that when you excite material and we are close to a metal surface, yeah, the energy immediately goes to the Fermi level of your metal, for instance. You quench your excited state and it's completely dead. So we had to lift it up. We put some legs to lift it up a little bit, and then they spin beautiful, and we could make this windmill park at the nanoscale. So this is our windmills in Holland. These are the windmills that we assemble. We have a surface or a monolayer, and all these windmills spin in the same direction. When we hit with light, when the light is off, they don't do anything. So we can now use them to change the surface. For instance, a droplet, we can contract and expand droplets and those kind of things. Now this Nano Car, and you have seen this picture maybe before. My students were jealous. I tell always this story because this really happened. My students, we are not in an engineering school, and in the engineering schools, what they do is they build these cars, maybe also in Sweden, with their solar powered, and they always can go to Australia for this car race. And my students were a bit jealous and said, can we also go to Australia? And so I said, we cannot do this because we are not engineers. But we can try to build a Nano Car, so it's a four-wheel drive Nano Car. It's 1 billion times smaller, as you can see. And I said, if you succeed, you can go to a conference in Australia. So we built here the frame, we built the 4 wheels. Yeah? It was not about building a Nano Car, of course. That's a gadget. What is the fundamental scientific question? The fundamental scientific question is how to convert rotary motion into translational motion. And can we see movement of single molecules? That is the question. And so this is what we did, and you see here a single Nano Car moving over a surface. We excite it with a scanning tunneling microscope tip. Now, when you have this, remind you of the movement in your muscles in your body, This is not the movement of a car. These are soft materials, and probably more realistic, and this is built based on theoretical calculations. This is the kind of movement of our cars. So it walks over the filaments or the surface. So finally, in your body, a lot of these motors, like the ATPase and all these motors, they are powered by chemical energy, by catalytic reactions. And as my group has been working on catalysis for so many years, I want to finish with an example of movement induced by catalysis. And so the whole idea was that you build a kind of a submarine or so. And then the question is, if you would like to have this in the body, what would be the fuel? And we thought, sugar, there's plenty of sugar. So we built carbon nanotube. We make carbon nanotubes, we functionalize them and put on 2 catalysts, and these catalysts work in concert to convert sugar. And what you see here is a tiny spider of these nanotubes because they tend to aggregate. We don't know yet how to control this precisely, but you see these bubbles, and these are the oxygen bubbles, and we know sugar is oxidized, it's converted. But as long as there is sugar, you see they find their path. They move autonomously. I don't touch it. This is just taken under the microscope. Some don't move because they're stuck, but these move like tiny spiders. So maybe in the future we can build roving sensors or delivery systems for drugs or micro-nano robots, but this is far distant from now. Maybe, we don't know. And maybe based on other systems than we have here. But once you know how to move, you can run fast. So will we have the fantastic voyage of Asimov in the future? I'm not sure, because it might be science fiction. But the best way to predict the future is to invent it. And this is what all the people here, and we all as an academic community together, are trying to do, to bring the future forward by inventing and by pushing the frontiers of human knowledge. So, to summarize the art of building Spore, we program molecules, we make responsive and adaptive functions, try to mimic some of the functions that Mother Nature used, but then in artificial materials, to induce motion and get to dynamic systems. So starting with Mother Nature, I talked about switches and motors. The perspective is responsive materials, as I mentioned several times now, self-healing materials, maybe delivery systems, precision therapeutics, I mentioned examples there, roving sensors or soft robotics in the far future. But once again, the message is we can control motion. And this is now a community that opened up a field, I think, where you will see a lot of fantastic voyages in the future. With that, I want to acknowledge my current group, or the group of the last year. They come from 14 different nations. We enjoy very much to work in teams. These are the young stars that make it all possible that I can tell the story here today. The funding agencies, of course, and particularly the Dutch Science Foundation and the European Research Council, which I'm very grateful to. But to close my lecture, the generations of students and co-workers that have in the past decades been working on these challenging topics, I'm extremely indebted to them. And I don't want I have not the time to show you 10 pages of names. Sorry, Fraser, but they are there. But I acknowledge them because we all know, all three of us know, we stand on the shoulders of our students that make it all possible. And I'm really indebted, and I feel very privileged every day to work with the young stars, the students, co-workers in my laboratories. Of course, the supporting staff, the colleagues in our laboratories, our institute and our university, the collaborations all over the world, the synthetic community, the supramolecular community, and the molecular machines community. And particularly I want to thank Fraser at this moment because he was mentioned Colorado. And for a couple of years now, we have this fantastic conference at 3,000 meters in Telluride on molecular machines. And coming from a country which is so flat, and 40% is below sea, our laboratory is in an area at Groningen which is below sea level, to go to 3,000 meters to discuss this beautiful future of our sciences, and with these fantastic colleagues, has elevated my academic career a lot. Thank you so much, Fraser.
Thank you so much, Ben. Fantastic lecture and also a great future for all of us to work in this field. Unfortunately, the Vice-Chancellor is waiting with the lunch, so I don't want to be later. I guess we will be yelled at otherwise. So please save your questions. Some of you will meet the laureates later and ask the questions then. I ask you all to help me thank the laureates for their fantastic lecture and also for the very bright future of us. Thank you very much.